Okay, guys. Today, inshallah, we're gonna continue what we started yesterday. So yesterday we started to talk about the cell cycle, and we said actually the cell go through some changes within, some changes periodically from the moment it produced from a cell division till it reaches a new cell division. And we said we during this cell cycle, the cell grow, develop, and then divide. And we classified the cell cycle into two major phases or two major steps: the interphase and the cell division, or the mitotic phase. We said that the interphase, it is the most cell cycle, or the most time the cell spend in its life in this phase, in which the cell go through many changes to be ready for the cell division, which means the interphase is not a part of the division, it's just a kind of preparation for the division. And we said the interface is going to be divided into three stages, which are G1, S, and G2. G1 is the rapid growth and the replication of organelles. That means the cell already make like double the content of the cell. Each organelle will have another copy, and the cytoplasm will be double amount, and so on. Then the S stage, in which the cell gonna replicate its DNA. So instead of single chromosomes that is look like thread-like structure, it gonna be like X shape, or we call them sister chromatids, that are attached by centromeres. So like this, we double the amount of the DNA, but still the same number of chromosomes, no change in the number of chromosomes, okay? And then G2, the cell already reaches the maximum size. That means it reaches already the maximum ratio between the surface area and the volume. And now the cell is ready for division. That's what we studied yesterday. So anyone have any question regarding to what we studied yesterday, guys? Anyone have any question?
No.
So actually, that's what we said. We said that actually cycle mean group of changes that happen in certain periods of time. And we said that the cell passed by some changes, growth, develop, and division. And we said that cell cycle is going to divided into two major parts. All this blue part represent the interphase, the most life cycle, while this part, the yellow part, that represent the cell division or the mitotic phase. Then we said that the interphase is divided into G1, S, and G2. As we said, G1, the rapid growth and the replication of organelles, the S stage, in which DNA is going to be replicated, and G2, that the maximum size and the cell now is ready for division. Here, that's what we stopped at last time, or yesterday. We just explained that that's how the chromosome will look like after replication. So we call these duplicated chromosomes, which are made of two sister chromatids, okay? So that means is counted as one, but the amount of DNA inside this duplicated chromosome is double the amount of the DNA which is already found in a single chromosome before replication. Okay, so today, inshallah, we're gonna pass to the main part here, or the other part of the cell cycle, which is mitosis, or the mitotic division. So actually, the mitotic division also is gonna be divided into two major phases, or two major steps: the mitosis and cytokinesis. Okay, so here everyone pay attention with me to this part, just to make a kind of summary for what we explained. We said that, okay, cell cycle will divide it into two major categories, as we said: interphase, which we already studied yesterday, and we know this interphase is divided into G1, S, and G2, and that's already done yesterday. Okay, we said what happened in G1. Once the cell in G2, it's ready for the next step, which is the division. We call this mitotic phase. So actually, the mitotic phase is divided also into two steps. Okay, what these two steps? It's divided into mitosis. That's okay. Let's try mitosis and cytokinesis. Okay. So what difference between them? Actually, mitosis, it means, okay, mitosis, it means the division of nucleus. Only so the mitosis, it's a kind of division in which the nucleus of the cell will divide. So that means after mitosis, we are gonna have a cell with two nuclei. Yes or no? Okay.
Yes.
The nucleus will divide. We are going to have two nuclei, and of course that is abnormal case. The cell is not, or the cell can't have two nuclei. Okay, because we know that the nucleus is the master of the cell. So I can't work, or the cell can't have two masters. So that can't happen here. What should happen? The cell directly pass to the next step, cytokinesis, which means division of cytoplasm. So in this case, or in this step, we are gonna complete the division. We're gonna divide the cytoplasm and what it's contained. So we're gonna have 2 new cells. You get the idea, guys? Okay. Is it clear?
Yes.
Yes.
So like this, okay, that's mean already the cell division happened in 2 steps. The mitotic phase, that the phase in which the cell will divide, but the cell not divide like once. No, actually it divide into 2 major steps: mitosis, which means the division of nucleus, okay, and cytokinesis, which means the division of the cytoplasm and its content of organelles. Okay. So our main part here, or our lesson today, we're gonna talk about how mitosis works and how cytokinesis. So mitosis is the division of nucleus. Nucleus also, as it contains the DNA in form of chromosomes, and now the chromosomes are replicated, we must work in a technique that ensure that we could divide this chromosomes to return back to the normal state in which single chromosomes in each new cell without any mistake. So here, that's why the mitosis doesn't just happen also as one step. It happened in 4 phases or in 4 steps. So it happened in 4 steps. We call them prophase, metaphase, anaphase, and last thing, telophase. Okay, guys. So that means already the nucleus divide in 4 steps. Each step has some changes that happen in. So these steps are prophase, metaphase, anaphase, telophase. So let's go through each one in detail and see what happens. Okay, just to clarify the idea for you. So here we're gonna find, back to our presentation. So we see that during mitosis, the content of the nucleus divides. So it means mitosis means the division of nucleus only. That's mean during the mitosis, the nucleus will divide to form 2 new or 2 identical nuclei. The cell will contain 2 nuclei, as you can see here. After all this is mitosis. After mitosis is done, I have 1 cell with 2 nuclei. Could you see it, guys?
Yes.
Yes.
Yeah, and we said that is abnormal situation. The 1 cell cannot have 2 nuclei. So in this case, okay, we must divide, okay, the entire cell, or divide the cytoplasm. That is the second step, which is cytokinesis. Okay. So here we're gonna find. Let's see how the division of nucleus happen, or how mitosis happen. So we said mitosis actually happen in 4 phases, or 4 major changes, which are the following. First one is prophase. So that's the first step. What happened in prophase? Let's look carefully here. You could observe now how the cell look like. So to be sure, okay, or to just be able to compare the changes, we must remember how the cell already looks like before starting the division. So to just remember, we're gonna draw a cell as usual. So here, actually, the cell supposed to be look like that.
Mr.
Okay, guys. Yes. Yep.
Mr.
Yes. I hear you. What do you want?
Mr.
Yes.
مستر بتهيألي مالك عندها مشكلة يعني هي مش سامعة بتهيألي.
Okay, so the others can hear me, yes?
Yes.
Okay, so that is the normal shape of the cell, okay? Actually, that represents the nuclear envelope or the nuclear membrane. This one represents the nuclear membrane or nuclear envelope. And that what the red material inside that look like a spaghetti dish, that represents the chromatine, which are tangled chromosome, as we said yesterday. Here, that this black spot is the nucleolus, and the green part, that is the centrosome or the centrioles, okay? So again, let's find here. Just a minute, please, guys. Sorry. Okay, so here we could find that is the nuclear membrane, and here that's chromatin. That means chromosomes already are tangled together to make like a spaghetti dish. This black spot, that's nucleolus, and here that's centrioles. Okay, or centrosome, and of course this black spot that represents the cytoplasm. So here let's compare what are the changes that already happened to be like that. Okay, so guys, let's compare. First, we're gonna find here what happens to the nuclear membrane. Look here, what happens to the, what do you observe?
Breaks down.
It breaks down, excellent. So the nuclear membrane start to breaks down, start to disappear, okay? So that's mean the chromatin inside here will not be trapped anymore, yes or no? It will get more space because already the border which surrounds it will be disappear. So, or will disappear. So it will take more space. So as it take more space, the chromosomes will not be obligated to be tangled around each other. They are gonna take more space. So each chromosome will condense, will appear separately, as we can see here. Each chromosome is alone. Can you see it?
Yes.
Okay. Yeah. So the first change happened that nuclear membrane disappeared, so DNA or replicated chromosomes will condense. Okay, so they will appear clearly. Okay. Here, what about the nucleus? Can you see the nucleus here?
No.
No. So nucleolus also disappear. Okay. So the nucleolus disappear, and then we are gonna find that the centrioles or the central here, the centrioles, each centriole start to migrate to one side of the cell. That means one will go here and the other one will go to the other side and start to form this spindle fibers. Okay, just like threads. Okay, is it clear? This guys, these green parts that are the
Mister, what is the green part?
what?
What is the green part?
Green part that they are centrioles. Centrioles are organelles found in animal cell in which they form the spindle fibers during the cell division. This one, we call this.
Okay. And they form the fibers?
Yes, centriole. Okay, we have two centrioles. Each one pass to one side during the division, and they start to form this spindle fiber. Okay.
Okay. Okay.
That's it. Okay. So that means there are 4 changes that happen during prophase. The nuclear membrane disappear, nucleolus disappear, then the chromosomes condense or the chromosomes appear clearly. Finally, centrioles migrate to each pole, forming spindle fibers. Is it hard like this, guys? Okay.
No. Okay.
No. So that is the longest phase, or that is the longest time of the, or that the longest phase during the division. Okay. So after already the cell do this, we pack to the second one, or we pass to the second stage, which is metaphase. So let's see what would happen in metaphase here. So here, that's our prophase. It will pass us to metaphase. So we call this, or that second one, which is metaphase. So could you guys tell me what you observe? What happened here? Okay. What happened to the chromosomes?
They are lined up.
They line up. They just stand on one line. So actually, what happened, guys? What you observed? How they line up like this? What makes them line up? Compare.
The fibers.
The fibers. Excellent. We could observe that the fibers which are already produced from the centrioles, the spindle fibers, they hold the chromosome from the 2 sides. Yes or no?
Yes. Yes.
So simply here we find that spindle fibers attach chromosome from the, sorry guys, sorry. So here we said that spindle fiber, they attach chromosomes from that centromere, the node which is found in the middle. Okay, this part. Okay, so each one is already attached from the middle, as we can see here. So we call this a centromere. and this spindle fiber keep pulling from the two sides until they make them align in one line at the center or the equator of the cell. That's why we put metaphase. The word meta in Latin means middle. So they line up at the middle of the cell. Okay, guys?
Okay.
Any other question here?
No, thank
No.
you.
So here, as we imagine, after a while, this spindle fibers, they keep moving until they catch each chromosome from its centromere, and then they keep pulling from the two sides until they arrange them in one line like that. So that's metaphase, okay? After metaphase, okay, we pass to the third one, which is the third step, which, anyone remember what's the name? The third phase?
Anaphase.
Anaphase.
So it's anaphase. So let's see what happened in anaphase. So as you can see, what's happening in anaphase? So I'm gonna clear the drawing. Okay. So what happens in anaphase, guys? What
They separate.
They separate.
Yes, they separate. Actually, the word ana in Latin means separation. So in anaphase, the spindle fibers are shrinking. Okay, they are shrinking. So as they shrink, they pull the replicated chromosome from the two sides. So after a while, it will separate or cut. You get the idea?
Yes.
Yes.
Yeah. So each chromosome is separated into two chromatids. Now each one will pass to one side because of the pull of the spindle fibers. So they move to opposite sides. So after a while, we are going to have some chromosomes in this side and some chromosomes in this side and the other chromosomes in the other side. Yes or no?
Yes.
Yes. Okay. Yes. So here, that's we call it anaphase. So anaphase, sister chromatids of replicated chromosomes will separate because spindle fibers are shortened or shrinking, pulling the chromosome from opposite sides, and the cell starts to elongate. We find that the cell is not like sphere; it starts to be like oval or long oval, as we can see now. Okay. So here. So that's here. Let's, as you can observe, you could observe the cell become more longer. Yes or no?
Yes.
Now we pass to the last step, which is telophase. Telo means the end. Okay. So I need you to look at the telophase and figure out what happened. Look what happened.
They divide.
Again, guys.
Divide.
Yep. Yes.
Nucleus
So each group of chromosomes, as we said, the chromosomes will pass to each side of the cell. So as they pass to each side of the cell, each group of chromosomes will be surrounded by a membrane, by nuclear membrane again. As you can see here, there's a nuclear membrane. Can you observe? Can you observe?
Oh, yes.
Yeah, so here nuclear membrane start to envelop or start to surround chromosomes one more time. And after a while, nucleolus will appear and the spindle fibers will disappear gradually. So we could find that at the end we have two nuclei inside the cell, yes or no?
Yes.
So the changes which happen in telophase is completely the opposite of?
Anaphase.
No.
Metaphase.
Which one? You can say 1, 2, 3 if you don't know, remember the names. That's one.
Prophase. Prophase.
Excellent.
One membrane disappear.
So telophase is completely the opposite of telophase. Why? Because in prophase, nuclear membrane disappear, nucleolus disappear, spindle fibers start to form. Here we do the opposite. The nuclear membrane forms around the chromosomes. The nucleolus start to appear, and also the spindle fibers start to break down. You get the idea? So they are completely the opposite. You get this?
Yes.
Yeah. So like that already we finished mitosis. That's mean by the end of telophase, I have one cell with two nuclei. Okay. So that is abnormal situation which caused the cell to pass to the last part of the division or the just next step, which is cytokinesis, okay, to divide the rest of the cytoplasm or the rest of the cell. Okay. So here like that. So we said that in cytokinesis, the cytoplasm will divide and form two new daughter cells. That means the cell just will split simply to form two new cells. Each cell is exact copy like the original cell, exactly the same, no difference except that it is smaller in size. Okay, guys? So here we said that. That's just a very quick revision for what we said here. Interphase, before this, guys. I need to find something here. In cytokinesis, we have a little difference between plant cell and animal cell. Let's see what the difference here. Okay, so that's here. Just a minute. So here we said that animal cell, and here plant cell. So let's see what difference. What difference actually? So simply we're gonna find that at the end of telophase we have a cell with two nuclei like that. Yes or no?
Yes.
Yes. So what happened? How the cytoplasm will divide? Simply we find that the cell membrane start to pinch inward. Okay, do you know what I mean by pinch inward? Go inside. Okay.
Okay.
For you guys, sorry. So I just need to erase this part only. Okay, so I try to erase this part. Actually can't. So do you imagine this part would be erased? Okay, I'll try to. That's one. This one would. Okay. So this part would be erased. Do you imagine?
Mister, what is this step called?
So what happened? The cell membrane. We said that the cell membrane, okay, pinch inward. That means start to go inside. This is the purple part. Is it? Can you see it? Okay.
Yes, Mister. This is in which step?
That's in cytokinesis. Already we divide. Already we finished telophase. That means the nucleus is divided, and we have two nuclei now. Okay, these two nuclei. Okay. Now
Yes,
we
okay.
need to divide the rest. We need to divide the cytoplasm. Yes or no?
Yes.
So the cell membrane start to pinch inward, making this part goes inside, and after a while, it will keep going to be deeper like this, to be like a groove. Okay, do you see it?
Okay. Yes.
Okay, we call this. Okay, we call this the cell furrow. Okay, the cell. Okay, the cell furrow. Okay, so this cell furrow keep increasing until they completely split the cell into two new cells. Is it clear, guys?
Yes.
That means the membrane keep going inside, going in until they completely touch each other and they be completely split. Okay, so like this, I will have two new cells exactly like the original, just they are smaller. Okay. Any other question?
Okay. No.
No. Back to the plant cell. Here we have a problem. What the problem? That means after already the telophase happened and I have the cell has two nuclei. Do you think that the cell is able to divide, or cytokinesis is able to happen like in animal cell? What do you think?
No.
The membrane is able to do the same like in animal cell?
No, because it
No.
has like
Excellent.
two.
No, because there is a cell wall. The cell wall is rigid, hard. Okay, it's like wood, so hardly to pinch inward like cell membrane. So in this case, what would happen? In this case, we find that there are some extensions, okay, that will be formed from the cell wall, and these extensions go through the cell until it form a new wall here. We call this cell plate. Okay, you got this, guys? Okay.
Yes.
So
Okay.
in this case, we said that extension of the cell wall, pieces of the cell wall, cellulose, will be formed inside the cytoplasm until it completely separate the cell into two new cells, and we call this extension the cell plate. So the only difference between plant cell and animal cell during cytokinesis, that plant cell forms cell plate while animal cell not, because animal cell lack cell wall. You get this, guys? Is it clear?
Yes.
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 Okay. So here like this, guys, we already finished. Unlike that, we have two cells, okay, or two new cells, which are exact copy like the original one, no difference between them, okay? So here, that's a kind of revise what we said. We said interphase happen in G1, S, and G2. G1 is just growth and cellular function organelle replication. S, the synthesis or replication of the DNA. S2, the cell reaches the maximum size. Dial here in mitotic phase, it happened in 2 steps, which are, who can remind me? Mitotic phase happen on 2 steps, which are? Who can tell me? Guys, girls. Mitotic phase happen in?
Mitosis.
Excellent, which means division of nucleus and
Cytokinesis.
Excellent, cytokinesis, which means division of cytoplasm. Brilliant, okay? So the idea now, what is the purpose? Why the cell should divide? Why the cell go all this through changing?
مثل some cells they get يعني
Because they stretch.
Yep, yes, we said that it divide once it reaches maximum size. The question, why the cell should divide or why the cell divides, okay? What the purpose? What?
For the organism to grow.
Yeah, excellent, to grow, because when you are young, of course, the number of cells in your body is not exactly the same number which is found in your body nowadays. Of course, you have more cells. So these more cells are already produced from pre-existed cells by division. So help in growth. What else? Do you know also replacement? Replacement, yes. Some animals, they have ability to replace missed parts, like in some kind of lizard, the sea star, and like that, okay? They could replace some missed parts, okay? So these new parts, which is gonna grow, they will grow from old cells, yes or no?
Yes.
In your body to repair your damaged tissues, repairing your, that means when you got wounded or you got cut in your skin or something, after a few days it's repaired. So how it's repaired? Actually, the cells keep dividing and they replace the damaged or lost cells. Yes or no?
Yes.
Yes. Beside that, also some animals or some unicellular organisms, they could reproduce asexually by division. Like for example, in some kinds of algae, some kinds of fungi, they just divide, and like this, like yeast fungus, for example, the yeast divide and produce new yeast. That's kind of reproduction, yes or no?
Yes.
Yes. So here like this, we find that the purpose of mitosis or the purpose of regular cellular division, just for 4 reasons. It's for growth, repair, replacement, and asexual reproduction in some living organisms, okay? So here, any hard question, guys, regarding to this part?
No, mister, thank you.
Okay, so like this here, we find that by this, we finished our lesson already. Okay, we just need you to try to answer the following question to check that you got the point. So what takes place during this stage of interphase? What happened during a stage of interphase, guys? Who can answer this question? You could raise your hand if you know the answer, please. Okay, Kenzie?
A.
DNA replication. Very good. Excellent. Okay. So DNA replication. Good. So let's see the next question. What are the 2 identical chromosomes that make up a duplicated chromosome called? That means each replicated chromosome is made of 2 identical DNA. We call them, is Judy?
D.
D, sister chromatids. Brilliant. They are attached to each other by centromere. Brilliant. Okay, the last question. Okay, during which phase of mitosis the duplicated chromosomes, okay, lined to the middle of the cell? So we say, let's see here, Nano?
C.
C. Oh, C, they just on the middle. We see that middle means in Latin. Okay, Fadia? Fadia?
B.
B, metaphase, okay? Meta means middle. Ana means separate. So anaphase is the 3rd phase, okay? After they already lined up at the middle in the 2nd phase, which is metaphase, they are gonna separate in the 3rd phase, okay? So any other question regarding to what we said, guys?
No, mister, thank you.
Okay, so that's the end of our lesson. And please, guys, try to use the weekend to gather all the information we explained or we studied during the 3 lessons, because starting from next week, inshallah, we're gonna meet you physically, or most of you, inshallah, and just have just more 3 weeks, and then we pass to our exams and so on. So don't think that we still have a long time. Actually, we cut half the way, so we need to be ready, okay, to the new things. So do your best, and thank you. See you next time.